This is an overview of an article entitled Ion Channel Dysfunctions in Dilated Cardiomyopathy in Limb-girdle Muscular Dystrophy, published in the March 2018 issue of Circulation, Genomic and Precision Medicine. Limb-girdle muscular dystrophy, or LGMD, is a rare disease of striated muscle with a number of genetic etiologies. A major cause of LGMD are mutations in the gene that encodes fukutin-related protein, or FKRP, which results in weakness of the pelvic or shoulder girdle musculature and, in some cases, can lead to dilated cardiomyopathy. Dilated cardiomyopathy, or DCM, is characterized by left ventricular dilation with loss of systolic contractile function. While DCM can be a primary disease, it can also occur in the context of other extracardiac syndromes, such as muscular dystrophies. Both LGMD and DCM can be associated with the development of life-threatening arrhythmias of the heart, but little is known about the way in which the heart electrically remodels in a way that can cause arrhythmia susceptibility. Recently, there have been rapid advancements in a novel in vitro tool for uncovering mechanisms of cardiac disease development. Patient-derived induced pluripotent stem cells offer a unique model for studying these unknown mechanisms. As outlined in a scientific statement from the American Heart Association, co-authored by the Councils on Genomic and Precision Medicine, Cardiovascular Disease in the Young, and Cardiovascular and Stroke Nursing in January 2018, the use of iPSCs in cardiovascular studies is expanding and can model cardiomyopathic and arrhythmic diseases alike. While specific methods can vary, in general, patient dermal fibroblasts, or blood-derived mononuclear cells, can be reprogrammed into pluripotent stem cells. Once validated, these cells can be guided down a cardiomyocyte differentiation pathway to become iPSC cardiomyocytes. As these cells theoretically can recapitulate the patient's genomic and cellular milieu, they can be used for cellular phenotype studies, molecular phenotyping, and in vitro drug therapy studies. In this study, skin biopsy was performed in a 51-year-old man of European descent with limb-girdle muscular dystrophy type 2I, who was wheelchair-bound due to skeletal muscle weakness. He harbored a homozygous FKRP variant, leucine 276 isoleucine. He also demonstrated DCM with a left ventricular ejection fraction of 30%. As a comparison, the iPSCs derived from this proband were compared to similarly derived iPSCs from 3 ostensibly healthy control individuals. Upon differentiation into iPSC cardiomyocytes, or iPSC-CMs, the electrical properties of the cells were explored, specifically the action potential, sodium current, L-type calcium current, delayed rectifier potassium currents, and transient outward potassium current. Finally, intracellular calcium signaling was explored. To get a global sense of the electrical properties of the patient iPSC-CMs, the authors first evaluated the action potentials of the cells. With whole cell patch clamp studies of the patient cells, also designated here as the DCM cells, compared to control cells, the authors observed a smaller action potential amplitude and lower maximum depolarization velocity, with a trend towards a shorter action potential duration. To explore the etiology of this electrical remodeling, the authors next evaluated the various individual ion channel currents constituting the cardiomyocyte action potential using voltage clamp recordings. Selected analyses of these channels are shown here. Starting with the upper left panel, the authors found that both peak and late sodium currents were significantly reduced in the DCM iPSC CMs. Not shown here, the authors also demonstrated changes in the sodium channel's recovery from inactivation, which was significantly slower in the DCM iPSC CMs. Correlated with this, there was a significant reduction in SCN5A expression, the gene whose product creates the channel responsible for the sodium current. In the upper right panel, the authors found a similar decrease in peak L-type calcium current in the DCM iPSC CMs. Overall, the L-type calcium channel activation and inactivation kinetics were similar, with a faster recovery from inactivation. There was no alteration in the expression levels of CACNA1C, the gene that encodes the ion channel imparting the L-type calcium current. In the lower two panels are the analyses of potassium channels, specifically the rapidly delayed rectifier current, or IKR. There was a marked reduction of the current in the DCM iPSC CMs, as well as reduction in KCNH2 expression, the gene encoding the relevant potassium channel. Not shown in this figure, the currents for both IKS and ITO were unchanged, despite reduced expression in the DCM iPSC CMs. Taken together, the authors concluded that these individual current changes underlie the observed action potential changes. In the final part of the work, intracellular calcium was analyzed after loading the cells with a fluorescent calcium indicator called fluo3 and imaged while beating. As seen in the upper left panel, DCM iPSC CMs demonstrated a reduced calcium transient amplitude during systole, as well as reduced resting levels of calcium in the cell during diastole. The authors concluded that, in addition to alterations in the electrical properties of the cells, there's also altered calcium homeostasis in the DCM iPSC CMs. Overall, the authors demonstrated that iPSC CMs from a patient with DCM secondary to LGMD are electrically abnormal. Specifically, these cells demonstrated reduced action potential amplitude and upstroke velocity. These alterations in the action potential were, at least in part, caused by reduced peak sodium and L-type calcium currents that were associated with reduced channel gene expression. Further, there was reduced delayed rectifier potassium current and altered calcium homeostasis. The authors concluded that LGMD and the secondary DCM are associated with electrical remodeling of the cardiac myocytes. They also concluded that iPSC CMs recapitulated many of the electrical changes associated with arrhythmogenesis, including ion channel conductance, channel kinetics, and intracellular calcium handling. Finally, they demonstrated that iPSC CMs can be used as a model for studying the pathogenesis of DCM and LGMD.